An important component of ultrasound imaging is sequencing. Sequencing is the process through which ultrasound pulses are transmitted and RF data are collected repeatedly by ultrasound systems. This slide provides an overview of how our sequencer works. We have a sequence table called a ray line table, which contains a sequence of transmit and receives in its rows. Each row corresponds to a line in the sequence. The sequencer goes over this table line by line, fires the transmit, receives the echo signals, and then moves on to the next line until the last line is reached. It then returns to the first line. The sequencer will repeat itself as long as the imaging is running on the system. The properties of the transmitted pulse and received data are not stored in the ray line table, but they are stored in separate tables. The columns of the ray line table contain only the data that is related to the sequence, such as its timing and amount of data to be collected. In the next couple of slides, I'm going to go over the most common types of ultrasound sequences that are used in conventional imaging. The first of these is the B-mode sequence. In the B-mode sequence, the tissue is scanned line by line by moving the center of aperture from one side of the transducer to the other side. Different lines are shown in the diagram by B1, B2 to B128 for the 128 scan lines. TB is the time that is required for acquiring a single line. This time depends on the depth of imaging and the speed of sound in the tissue. The total time that is required for scanning all the sequence is TB times the number of lines in the sequence. This is the frame rate of the B-mode sequence. The next sequence is compound B-mode or spatially compounded B-mode. In this sequence, B-mode frames are acquired at different angles and then combined to produce a better quality image. The figure shows three different angles. The sequencer acquires the scan lines at the first angle, one by one, scanning from one side of the transducer to the other side, then moves on to the next angle and then the next. If each angle is treated as a separate frame, the data can be pipelined and the image processing algorithm can always process the last three frames, which will be at three different angles. Using this method, the frame rate of the compound B-mode would be the same as the conventional B-mode, although there will be a very small lag. Pulse wave Doppler or PWD is used to monitor the blood velocity in blood vessels. In PWD mono, B-mode images are not acquired. Only a single scan line is repeatedly acquired at a set pulse repetition frequency or PRF. The T sub PW, which is the minimum time required to acquire the PW line, depends on the depth of the gate and speed of sound. T sub PW should be smaller than the pulse repetition period, and therefore the depth of the gate determines the maximum achievable PRF. In PWD duplex, B-mode images are also acquired at the same time as the PW lines. If the PRF of the Doppler is not very high, there will be sufficient time in between successive acquisitions of the PW lines, and in these intervals portions of the B-mode sequence can be acquired. For example, in the figure, two lines of B-mode are acquired each time. The sequence in this case will consist of a PW line, then B1 and B2, then the PW line again, then B3 and B4, and will go on until all the B-mode image is acquired. This sequence is usually called uninterrupted duplex because the Doppler sequence is not interrupted for the acquisition of the B-mode. If a higher PRF is desired for PWD in the duplex mode, there will not be enough time between the successive acquisitions of the PW line. In this case, in order to acquire parts of the B-mode image, the PW sequence needs to be interrupted. Therefore, the sequence is called interrupted duplex. After a number of PW lines, the sequence of PW lines is interrupted and a portion of the B-mode is acquired. Then the PW lines resume. This process is repeated until all the B-mode sequence is programmed. As can be seen from this figure, there will be gaps in the PWD data. These gaps are dealt with in the software. Another important sequence is the color Doppler sequence. Color Doppler is also used to monitor the blood flow in blood vessels. Color Doppler is usually acquired with live B-mode images at the same time. For color Doppler, the lines inside a region of interest, or ROI, need to be scanned at a certain PRF. However, unlike PWD, it is not necessary to scan the ROI continuously. A certain number of frames, called the ensemble, is enough. In order to achieve the desired PRF, the ROI is divided in smaller sectors consisting of fewer lines. These lines can be scanned in a sequence, while repeating this sequence will still meet the desired PRF. For example, in the diagram, the sector consists of 4 lines denoted by V1 to V4. The sequence is then repeated the desired number of times to complete the ensemble. Then the sequencer moves to the next sector inside the ROI and so forth until the entire ROI has been covered. Once done with the color sequence on the ROI, the sequencer will go on through a B-mode sequence going from B1 to B128 to cover the entire image. If PWD, color Doppler, and B-mode are acquired at the same time, we have a triplex sequence. If the PWD has a low PRF and the color PRF is an integer divisor of the PWD PRF, then there will be enough wait time in between the successive acquisitions of the PW lines, and in these wait times the color Doppler can be acquired synchronized with the PWD, and once all the color lines from the ROI are acquired, the B-mode lines can be fit in the wait time, one portion at a time, until the entire B-mode image is acquired. However, if the PWD PRF is too high, it needs to be interrupted in order to make room for the color Doppler and B-mode sequences. The B-mode sequence can be separated into smaller portions. However, the color Doppler has its own PRF, and until all the ensembles are acquired for one sector, the color Doppler sequence cannot be interrupted. As a result, the PWD sequence has to wait for longer periods for the color Doppler ensembles, and this creates longer gaps in the PWD and lowers the quality of the measurement. With this slide, I close my discussion on the conventional sequences and move on to discussing some details of our software and hardware architecture.